Hi, I'm Dr. Arshad. Today I'm going to teach you some key terms and important points that are frequently asked in exams. First is interleukin 1. Interleukin 1 is important mediator for necrosis. But is it programmed? No. Necrosis is never programmed. Other hand, a type of cell death is programmed called apoptosis. Apoptosis is programmed and it is simply caspase enzyme is remembered here because it is executioner one. That means it uh, maintain the function of cell leading to all mechanism important for finally death of cell, one cell or a group of cells. So it is apoptosis. Now what is annexin 5? Annexin 5 is a marker that is applied to check apoptosis. You know, when fibrolysosome formation happen, it leads to further apoptotic bodies which are like groups in groups or bunches. Annexin 5 act on them and you can identify the process of apoptosis there. It has very important role when we apply some drug, for example chemotherapeutic drug, for example in a tumor. When we want to see that is apoptosis happening or not by the action of drug, we can check by annexin 5 marker. So in simply by microscopy we can check that apoptosis is happening or not in the tissue by annexin 5. What is force, gen and mink? The three are product of gene. They are important for cell growth. What is AKT pathway? AKT pathway is important pathway for the hypertrophy. What is hypertrophy? When size of cell increases because of adaptation, it's called hypertrophy, and the pathway is AKT pathway. Here we discuss the three very important stain. The first one is Sudan. Sudan is for fat. Sudan is used to stain fat. One cosa is used to stain calcium, and Giemsa to stain bacteria. The four terminology: what is hyperplasia, what is hypertrophy, what is metaplasia, and what is dysplasia. We define it simply like hyperplasia is increase in number of cells, and what is hypertrophy? It is increase in size of cell. What is metaplasia? When one cell type is replaced by other cell type is called metaplasia. And what is dysplasia? Dysplasia is when there is a disordered cell growth, or when the cell growth pattern is disordered. It's called dysplasia. So which on the four condition is reversible and which one is partially reversible? Simply here hyperplasia, hypertrophy, metaplasia, all are the changes of the adaptation, and adaptation changes, so they can even change to the normal condition when the physiological demand meet according to the condition. So question asked is metaplasia is reversible or not? It is reversible. But is dysplasia reversible? It is partially reversible. How the reversibility is checked here? By quiescence of the irritant or the stimuli. If the stimuli is being stopped, then reversibility chances is here. So dysplasia is defined as partially reversible. APAF is very important because it binds to cytochrome C, and it is mitochondrial mechanism. Apoptosis has two types, extrinsic and intrinsic type. Extrinsic type is by death receptor or by death domain, whereas intrinsic is mitochondrial. It is because of release of cytochrome C and binding of a path to cytochrome C and activation of caspase. Now, one last point is myelin figure. What is myelin figure? Myelin figure is found in the cell and important to a study. What? Some changes in the cell. It is of the membrane origin, bonded vesicle like a structure in the microscopy. So what is the exact significance of myelin figure? I shall discuss in the next video.